The fourth and last type of biological molecule that we're going to discuss are the nucleic acids. So this deals with DNA and RNA. Clearly, DNA and RNA are also polymers, like most of the biological molecules that we've discussed, except for the lipids. They form long chains, but the individual subunits of DNA and RNA are made out of nucleotides, and we'll take a look at the structure of those nucleotides very, very soon. All right, so DNA and RNA have a lot of similarities, but they also have some differences. Let's list some of them. DNA tends to be double-stranded. DNA tends to stay in the nucleus. DNA has the following nitrogenous bases. It has A, T, G, and C. RNA doesn't have a T, it has a different one. And DNA has deoxyribose. For the sugar, for the pentose sugar, that's one of the components of it. RNA is single-stranded, usually. Not always, but usually. Most of the RNA we're going to talk about is mRNA. That's the major type of RNA, and that tends to be single-stranded. It leaves the nucleus. It has the bases A, U, G, and C, and its pentose sugar is ribose. All right, so let's talk about the nucleotide components. There's three components: pentose sugar, a nitrogenous base, and a phosphate. The pentose sugar is either going to be ribose or deoxyribose. And we draw the pentose sugar like this. Okay, so it's going to be a pentagon with an oxygen at the top here and then one antenna carbon sticking out. We're going to label this carbon 1. This is going to be carbon 2. This is 3. This one here at the corner is 4, and then the top one sticking out is 5. All right, and then depending on if there is a oxygen or hydroxyl on carbon 2, it makes it either ribose if there is one, or if there isn't one, it's deoxyribose. That's the one that's found in DNA. All right, so the nitrogenous bases are then, they're either going to be two-ring structures like the purines that we're going to see. They're going to look like this. And by the way, they have, they're fairly complex structures that we're going to just simplify like this. Or we have pyrimidines, which are actually smaller. They're just a single ring with 6 corners, like that. All right, so A and G are going to be the purines. They're the big ones. And then T or U and then C are going to be the pyrimidines. Oops. All right, and when DNA links together, when you form a double strand, you always have one purine, one of the big ones, linking up with one of the small ones, like that. And then finally, the last component of a nucleotide is phosphate. And phosphate is just simply a phosphorus and 4 oxygens. There's one double bond O, and then this last oxygen is connected to carbon 5 of the pentose. So that's going to be connected to the sugar up here. All right, so why don't we draw DNA, why don't we draw a nucleotide altogether? I'm just going to switch from this present view because it really messes with my, it makes a lot of lag happen in my pen, so I'm just going to exit from this. It's going to be a little smaller for you, but it's just going to work a lot better. I'll try to make this as big as possible here. All right, so let's draw some nucleotides, and maybe we'll even link them together. All right, I always start with the pentose, so I'm going to start with the deoxyribose. Let's say we're going to draw DNA right now, so I'm going to draw deoxyribose. All right, so I got my pentagon here with the oxygen on top. So there's oxygen in the ring, just like glucose has an oxygen, but this is a pentose, only 5 carbons total. Here's carbon 5, here's carbon 1. So here's 1, here's 5. All right, so that gets a phosphate on it. And I'm going to abbreviate the phosphate. I'm going to be a little bit lazy and just do this, because I don't want to draw that PO4 thing every time. So we know that that's what it is, but it's easier just to do that. All right, then we're going to stick a nitrogenous base on here. So I'm going to start with just putting, oh, why did it go black, Austin? I don't know why. Did I do this? Go blue again. All right, so we're going to do pyrimidine first. We're going to draw it like this. All right, I know my ring is kind of crooked, but that's the way it goes. All right, if we were going to draw another one, we'll put it right below the other one. So I'm lining these up as best as I can and do another one like this. And I'm going to try to line up my phosphates also, so they're right below each other. That makes it easier. This phosphate's then going to get connected to this carbon 3 right here. Okay, and the other phosphate on top is connected to carbon 5. Let me fix this black line to blue. All right, good to go. All right, so now I'm going to draw a purine. So this is a pyrimidine with only one ring. So this could be like, I don't know, T, let's say. Now I'm going to draw one that has the 2-ring one. All right, so it gets the pentagon first, like this. And then the hexagon attached to that. So on the other side, then we basically get the opposite type of molecule. So if we have a pyrimidine here, a small one, we'd get a big one over here like this. Oopsie. All right, except now they're going to be upside down. All right, so I'm drawing my pentose upside down, my deoxyribose. The oxygen is facing down now. And these line up here, and these guys are going to connect with each other with hydrogen bonds. There'll be 2 hydrogen bonds here that sort of, hold them together with the sort of medium amount of strength. All right, we'll put our phosphates on there. And then over here, we'd have, since we have a purine here, we'd have a pyrimidine over here. This. Sorry, my drawings are sloppy, but all right, so the phosphates are lined up again. Now this phosphate connects to this carbon 3. Here'd be carbon 1, 2, 3, and this is carbon 5 over here. All right, so this is, if this is T, this would be then A, and if this is A, this would be T. We could also, make one of these, we could say these guys are maybe G, and then this would be C, just to give it a little variety. But there's nothing that says that if there's a C here, there has to be an A here, or if there's a G, there has to be a T. These could be in any sequence. There could be 2 A's in a row, or 3 or more. It could be mixed up in any order. It depends on the gene. The gene gives the code for the order of these letters, and the gene is going to depend on what protein is needed. All right, so now we have double-stranded DNA. This would keep going down here. This would keep going up here. This would keep going here and here. So we have a polymer, but it's really 2 polymers sort of linked together, and we end up calling that the double helix. All right, so this is just fancy diagrams of everything we've talked about, showing the nitrogenous bases, the pentose, the phosphate groups. Notice that we have deoxyribose in DNA, we have ribose in RNA, and just the carbon 2 oxygen is missing on deoxyribose. That's about the only difference there. We list the purines here, the big ones. They have 2 rings. And we have the pyrimidines here, the small ones. They only have 1 ring each. And uracil is an oddball that's only in RNA. RNA does not have thymine, it has uracil instead. So the purpose of DNA really is something we should mention briefly. We're going to go into more detail on this later in the year. But the purpose of DNA is it's a recipe to make protein. So the diagram we have here really is something that scientists call the central dogma of biology, which is kind of a big deal apparently. Central dogma, my goodness. The central dogma basically says that DNA leads to RNA, which then leads to protein. DNA leads to RNA, leads to protein. DNA is not converted into RNA. It doesn't magically change into RNA. It is the recipe to make the messenger RNA. And then the messenger RNA has a copy of the recipe from the DNA in order for the ribosomes to make the correct protein. So the DNA, as we said, is in the nucleus. It stays in the nucleus. It gets transcribed. That means a copy of the DNA of the gene is made in the form of mRNA. The mRNA is then able to venture outside of the nucleus through these pores, is able to leave. And then out in the cytoplasm, it meets up with a ribosome where it connects to this ribosome and then feeds the ribosome the message that it requires to make the protein. And the protein is made out of individual amino acids. These amino acids are linked together by the ribosome with dehydration synthesis. And the way that the ribosome knows which amino acid, which of the 20 it has to put in here in the right sequence, that information is given to the ribosome with the mRNA. So here we have our mRNA feeding through, it's going in this direction, and it's basically cranking out this polypeptide, this protein, with that message. Interestingly, a ribosome is also made out of RNA. It's not actually made out of protein, it's made out of rRNA. So ribosomes are made out of RNA also, which tells us that RNA can act like genetic material. It can hold a message or a recipe, but it can also act kind of like an enzyme or like a protein. So that's something we're going to run across again in this class, where RNA functions in a number of different ways. It's kind of a Swiss Army knife of molecules. The final thing I'll just mention briefly is that DNA will replicate. It'll copy itself when it's time for cell division. Right before cell division, right before mitosis, the DNA has to make a copy. So basically there's an enzyme that's called DNA polymerase. Notice it has the word polymer in it, so it just basically is an enzyme ACE that makes a polymer, and it links the nucleotides together in the correct fashion to make a second strand of DNA. So the orange here is the new strand where DNA polymerase links up the correct nucleotide, the matching one. We say it's the complementary one. For example, this C gets a G. This A over here is going to get a T. This G is going to get a C over here. And then 2 new double-stranded pieces of DNA are going to form. That's going to go to one cell, and this is going to go to the other cell. All right, so the 2 daughter cells that form in mitosis, in cell division, each get a complete new set of these chromosomes thanks to replication, which is copying DNA. All right, we'll talk about this in much more detail later in the class. I just wanted to plant some seeds here with that.